Welcome back. Before the break, we looked at the cell cycle and we said that cell division was very important for growth and for replacing cells that had been damaged. But mitosis is extremely important for unicellular organisms and how they actually reproduce themselves. Now, you know that when you were made, you started off as an egg, which was fertilized by a sperm cell, and that single cell that was produced was called a zygote. And you grew from that zygote into a huge multicellular organism. Now, for cells that are organisms that are unicellular, the whole organism is made up of one cell. Mitosis is vitally important because that is how those organisms reproduce. So bacteria and unicellular protistans, such as amoeba, reproduce by a process that is basically mitosis. This mode of reproduction is asexual and it is called binary fission. The term binary refers to two and fission means to split or to move apart. So one single organism is going to, through the process of binary fission, split into two organisms. It is an asexual mode of reproduction because only one parent is involved. Only one organism is involved, not two, as happens in sexual reproduction. There is no combination of genetic material as what happened with sexual reproduction. We can predict, therefore, that the outcome of binary fission will be two cells which are identical to each other as well as being identical to the parent. Organisms which are identical to each other are known as clones. So binary fission produces clones of the parent cell, and this is very important in organisms such as amoeba, which belongs to the kingdom Protista. We can see on this photograph here that we have one organism, one example of one part of the amoeba splitting in half, and this is the process of binary fission. The one cell simply splits in half. For prokaryotes, this form of cell division, binary fission, reproduction, is very, very important. Remember that prokaryotes are organisms that do not have a membrane-bound nucleus containing DNA. Bacteria are prokaryotic organisms, and like other prokaryotes, all of their genes, or most of their genes, are held on one chromosome. And so if we had a picture or a diagram of what this looks like, we've got the nuclear material here with their genes on their DNA on one particular chromosome. The single bacterial chromosome just is one single DNA molecule, and it has a couple of proteins that actually just hold it together. Now, when that single chromosome is actually stretched out, it's probably about 500 times longer than the bacterial cell itself, but it's highly coiled up and packed away inside the cell. Prokaryotic organisms undergo binary fission when they reproduce. And if we're going to make two organisms out of one cell, we need to take that DNA and we need to replicate it, which means that we need to copy it precisely. It would be impossible to create a new organism where the new cell only had half of the DNA. So we need to actually take that whole chromosome and we need to replicate it. And the chromosome replicates exactly in the same way as your own DNA replicates itself in your cells just before cell division. At the end of the cell cycle's interphase, DNA has to replicate and the chromosome has to be copied, and a complete copy in precise and absolutely correct in every way has to be made. And once this has happened, we're ready to go into the process of binary fission. So it starts off where we have our bacterial cell with its single chromosome. The cell grows and the DNA replicates itself or makes that exact carbon copy of itself. Then, certain areas of the chromosome where replication originated or started have to be joined up. This is because it's a circular chromosome, not like your chromosomes, which are linear. But once the chromosome has been copied, those little circles have to join up again. And now we've got a bacterial cell that's got double the amount of DNA in it. It's got a copy of its own DNA. And the two chromosomes move to the opposite ends of the cell. They split from each other and they move apart. Now the interesting thing that happens is the whole cell begins to split in half. The plasma membrane grows inwards and a cell wall is deposited down the middle of that cell, and the cell wall moves outwards until it separates the cells from each other, and two identical cells have been produced in this way. It's very interesting to think about certain antibiotics that are medicines or chemicals that you take to kill bacteria actually prevent the growing of that cell wall. So when you get a bacterial infection, the cells cannot replicate because they cannot make the cell wall. After the break, we're going to look very much more in a lot more detail at the process of DNA replication.